sure I switch it. Oh, okay, yeah. It was perfect timing. You don't want to stare on this picture all day here.
So what's his name?
His name is Bernd Schüssler from Gothenburg, and so this is in the Swedish National. It was about a month ago. All right, let's see if I find my presentation there. Okay. All right. All right. Well, thanks, Andika, for that nice introduction and for giving me the opportunity to be the first presenter in this series. The tennis, I'm still going to keep my day job, I think. It's not very lucrative in my age group. So you talk about math, there will be very little math today. It will be, the only math is the 4 in the title, I guess. And what you have in mind for 4D is probably very different from what I meant in my title, but we'll see. So anyway, so our group is very interested in better understanding the regulation of our genome. And to that end, we have developed a set of techniques that allows us to look at various aspects of gene expression. Things like the synthesis of RNA, RNA synthesis rates, the turnover rates of RNA. We also can look at the rate of transcription elongation down genes. And we can also get some information about promoter usage and enhancer usage in a genome-wide fashion in the cells of interest. So what I'm going to do today is I'm going to introduce those, introduce these techniques for you, give you some examples, and I'll give you 3 short stories with these different techniques. And if you find that these techniques may be useful for you, let me know and we can figure out a way to collaborate. Okay, so if you think of transcription, forward. I think of transcription a little bit like a musical score. There's a set of instruction that needs to be read in a certain order. So you have the primary sequence, but then you have instructions for the musician how to play, if you play it soft or loud or fast or slow. And these are sort of the epigenetic marks that tell cells whether to really express a gene or repress a gene. So when you think of transcription, obviously the binding of a transcription factor to the promoter or enhancer is obviously very important for regulating transcription. And the transcription factor binding and enhancer binding really drives the rate of RNA synthesis, which is kind of 1D of transcription or gene expression. But then you have what happens after the RNA is made. You have a time component where things happen to the RNA. It's being processed, the introns are removed, you have maturation of the RNA, and then it's subjected to degradation at some point. And each RNA has different set points, so they degrade at different rates. So we're very interested in trying to understand this and to measure this. Another rate that's important is the rate of elongation, and this makes a lot of, it's important for large genes. So some genes in our genome is like 1 megabase long. A little change in the rate of elongation could make a big difference in how fast that transcript is made. And then finally, we're obviously very interested in how these 3 aspects of RNA or gene regulation is regulated. And we have focused with one of our techniques on looking at enhancer element usage and promoter usage. Now, when we think of many types of diseases have defects in regulation of genes. And if you have defects in regulation of genes, then the symphony doesn't sound very good. I don't know what this would sound like. But anyway, so we have developed these 4 techniques, and I will describe them to you today. The first one is called Bru-Seq. It gives us a handle of the rate of transcription genome-wide. The Bru-Seq, here we age the RNA to any age we would like it to be, and that allows us to look at splicing kinetics and also the turnover of RNA genome-wide. The BruDRB-seq technique allows us to measure the rate of transcription elongation on many thousands of genes. And finally, the BruUV-Seq allows us to enhance for reads that's coming off of promoter, transcription start site, as well as enhancers. And the basis of these techniques is the labeling of the newly made RNA with bromouridine. So the first step for all of these techniques is to label the newly made RNA. So bromouridine enters cells, it gets incorporated into the newly made RNA. The next step is to isolate total RNA from your sample and fish out the BRU labeled RNA specifically using antibodies, magnetic beads. So we capture this newly made RNA that was labeled, we get rid of all the other RNA, and then we prepare libraries, and then we go over to Bob and he sequenced for us, and then we get a lot of data back and we analyze that. And we have, since our data is a little different from regular RNA-seq data, we have spent quite a bit of time to develop analysis tools specifically for our data. So we have a good pipeline now for this. All right, so to just give you a kind of flavor of what we are seeing with this technique. So this is mapping of the newly made RNA, and we're looking at one particular gene here in the genome, it's MAD4, and you see the map. Let's see if I know. Okay, so you see the map of the gene here. You have an exon, intron, exon, intron, and so forth. And here's the 3' UTR. And you can see the newly made RNA. You get a signal throughout the gene. Even you get some signal past the termination site. You often for genes see this divergent upstream transcript. So when you see signal on the upper strand, that means transcription goes from left to right. If it's on the bottom strand, it's transcription going on the other strand from the right to the left. Now, if we, another thing that people notice, obviously, is this choppiness, and this is sort of inherent from making the libraries. It's a PCR reaction of millions of pieces, and as you know, when you do PCR, some primers, some sequences amplify better. So this is very systematic. We see this every time we do it. We see the same peak. If you now age this RNA, so we label it for 30 minutes, we wash out the BRU label, we chase it with uridine, and we do this for 6 hours. So we look at that RNA we made 6 hours ago. What does it look when we pull it out 6 hours after it's made? And this is this Bru-Seq, which is color coded. So every time we look at something red, it's the aged RNA. So obviously, when we aged it, all the introns are taken out, and we're left with introns of UTR. And these sequences here are very unstable, and we don't really see them. So now the relationship with the signal at 6 hours over the exon to the signal over the whole gene at time 0 gives us an indication of how stable this particular RNA is. So you get a relative stability this way. So this is a fairly regularly middle of the pack kind of stability transcript. If you were to look at the FOS transcript, that's one of the most unstable transcripts, and you're hard pressed to even see the red, the signal at 6 hours. It's down here. So this gene is actually transcribed at a fairly high rate, but the transcript is so unstable it just disappears. So when you do steady state RNA, you probably don't see FOS very often, but with the Bru-Seq data, we see the gene is actually fairly active. Yes? Yes, you sum up all the reads from the beginning to the end of the gene. And then you can order, this is in RPKM values, reads per 1,000 base per million reads. So you can compare different genes with each other, and this really describes the transcription rate we're looking at in the cells. So a lot of people do shRNA, and when we do shRNA for a target gene, we can actually see that it's targeting the stability of the transcript. It doesn't affect the transcription rate of the gene. And so here's an example. We were knocking down PARP, and this is just the scrambled shRNA. You can see the 6-hour-old RNA for PARP. It's very stable. You see all the exons here, but if you target PARP with shRNA, you can see stability has drastically been reduced. The blue here is the nascent RNA sequencing. So the transcription rate of the gene didn't change, but the stability of the transcript changed. So this is confirming that the knockdown was good. And this is going to be true for genes that you look at with microRNA and so forth. It's going to target the stability of the transcript, not the rate of transcription. Now, if you follow, so in this example, I did 0 hour, and then I aged it to 6 hours. What happens if you take samples in between? You do time course and look at what happens to the introns and the splicing over time. And this is a gene, it's a large gene, it's like 300 kb long, and we're going to follow. So this is just the rate, the nascent RNA transcription of the gene. You see signals throughout the gene. And now we're going to chase it with uridine for different periods of time. So we're going to age it and to see what happens. So 2 hours, 4 hours, 6 hours. And this is an unusual gene in that if you follow this intron, there's very little happening over time for this intron. It's still retained at 6 hours, while the neighboring introns are going away pretty quickly. So we've done this genome-wide now, and we find genes where you have multiple introns, and the rate of intron removal or the signal in the introns are very different. And when you go back to the original reads, we can determine whether this intron was not spliced or if it was spliced, but for some reason that intron escaped degradation. And so we have examples throughout the genome of both of those cases. Certain introns, a number of introns are very poorly spliced, while some are spliced, but the intron, for whatever reason, is retained. It doesn't turn over. So potentially, there might be some non-coding RNA function in these introns, and that would be interesting to follow up on. Now, we can also use an inhibitor of general splicing. This is a drug called Meayamycin, and this is just, again, the normal rate of transcription in control. If you treat cells for 6 hours with Meayamycin and block splicing, genes can still transcribe. And this was a little bit of a surprise to us. We thought that in order for RNA polymerase to elongate down the genes and synthesize RNA, it needs to have things behind it spliced correctly. But obviously that's not the case. You get pretty much the same amount of transcription going on, even though splicing is inhibited. It's a little bit less of transcription reaching the end of the gene, perhaps. Now, if we chase this RNA over time to see if splicing takes place, there's really nothing taking place. So this drug very effectively blocks splicing. You can maybe notice that it seems like these transcripts are being degraded from the end here, and that's something we're currently investigating. Now, I'm just going to show you one more thing. This is data we got this morning, so I don't know the biology of this, but it's just another thing to what our techniques can do. And since we're looking at strand-specific data, so this is the data we did. We did an experiment with control and hypoxia-treated cells. And we know that HIF1 alpha is a major target for hypoxia. We didn't know much about the transcription of this gene, but this is under control condition. This gene is nicely transcribed. We get a nice read from the direction of the gene, and nothing on the other strand. But if we treat with hypoxia for 24 hours, this is what we see. We still get reads from the plus strand, the sense strand. All of a sudden, we see this massive induction of this antisense in the gene. And this is a Ewing sarcoma cell line. So this is a collaboration, and this is Beth Lauer's lab, and they were interested in hypoxia and Ewing sarcoma. And we haven't gone over the data yet. She hasn't seen it yet. This is just an example of the things we see, and there might be a nice explanation for it. Okay, so I had a summer student, Haley Lefkowski, that decided to look at all the data we have accumulated over these last 3 years when we've done the sequencing. So we have now 13 cell lines where we've done both Bru-Seq and BruChase-Seq. So we have synthesis and stability in these 13 cell lines. And she was going to look at, she took, as an example, looked at 29 DNA repair genes across these cell lines. What we noticed when we look across cell lines for different genes, we can see that one cell type may synthesize a lot of a specific gene, but the transcript is unstable. Another cell line for the same gene may not synthesize much, but the stability is very different. So each cell line has ways to change the set point for synthesis and stability. And there's probably a lot of interesting regulation here. But as a test case, she decided to look at these 29 DNA repair genes. And just to get a little bit of biology, what this DNA repair pathway is doing. This is called nucleotide excision repair. This is a crucial repair system when you go out in the sun and get lesions from UV damage. So these are these pyrimidine dimers. These are helix distortion, and they block transcription, and they can also interfere with replication. And this system has developed over time to deal with these lesions. So in the first step, you have recognition of the lesion. This complex looks at lesions all over the genome. This is global genomic nucleotide excision repair. These factors have the help of the RNA polymerase getting stuck and recruiting these factors so that this lesion can be removed in front of the polymerase, and supposedly you can resume transcription. This is called transcription-coupled nucleotide excision repair. And then you have factors in preparing this lesion for incision, and then finally you have a resynthesis and ligation. So just to illustrate this fact that cell lines behave very differently for different genes. So here are two, these are pancreatic cancer lines. This is the XPA gene. It's synthesized to the same degree, but you can appreciate when we do the BruChase-Seq that the stability of these transcripts are very, very different. So there's very, very different regulation, post-transcriptional regulation in these cells for this particular transcript. And similar for this other XPC, very different levels of stability. So we decided to try to look across these cell lines and form these heat maps. And so here are all the 29 genes, and here are the 13 cell lines. And it's color-coded, so the more yellow, that means it's more synthesized, and black, it's very little synthesized. So you get these complex pictures, and you can maybe pick up, oh, this gene is very poorly synthesized among all these genes. You can do the same for stability, and you get this kind of pattern, and you can stare at this for a while. Actually, here, the RPA that was very poorly synthesized, actually, that transcript is very stable. this analysis. So then we thought, okay, is there a way for us to kind of grab all the data across these different lines here so we can get a gene score? And when we do that, so we look at the expression of these 29 different genes across the 13 cell lines to look at which gene is the mostly synthesized among them. And here we see the RPA, as I showed before, very poorly synthesized, and you can rank them. And the same for stability, you can rank them. And here you can see the RPA switching from poorly synthesized, but it's very stable. So that's how it maintains a homeostasis of expression. You can do the same for cell lines. And the ultimate goal of this is just a test case, but when we have a number of cancer lines and we get this sort of cell score, we are hoping to determine a particular cell line may be very vulnerable to particular treatment because the cell score in a particular pathway is very low. And so those could generate hypotheses that we can test both in cell culture and in xenograft. So that's sort of the ultimate goal of this. We noticed one thing. So one little biology part here that we put together in this paper we submitted is this ERCC6 gene. It's a fairly unstable, one of the most unstable transcripts, but it's also a very large gene. And this gene codes for this protein ESB. This is very important for survival of cells with transcription coupled repair. So if you have a gene producing a very unstable transcript, and this gene is actually very large, so it could be hit by these transcription inhibiting lesions. It might act as a dosimeter. That's sort of our hypothesis. So under low stress, so low UV doses, the gene, which is very large, is mostly undamaged. It can produce the RNA, make the protein, and you can survive. But if the dose is higher, since the gene is fairly large, it's actually 3 times the size of the average size of genes in the genome, it becomes sort of a target for inactivation. And any RNA that was previously done or made is very unstable. It will go away. The protein will not be made, and now you have a switch into survival or to cell death. So this is the hypothesis we put forth, that it may be a dosimeter really regulates cell death after UV. All right, the second story is looking at the rate of elongation of RNA polymerase II down genes. And to do this, we employed a chemical that's called BruDRB. So normally, the RNA polymerase, when it initiates, it lands on the promoter, and it waits. Often it's arrested in a promoter proximal place, and then it waits for these kinases to phosphorylate the C-terminal domain, and that allows it to get into elongation mode. There's a drug called DRB that inhibits this class of kinases. So now you can efficiently block the polymerase at the promoter, and it can't elongate. So just to illustrate this, in order to look at transcription and measure elongation rate, it's really impossible under normal conditions because each gene will just fire asynchronously and go down the gene. But with the help of DRB, we can block the polymerases at the promoters. And now when we wash out DRB, we can allow these to synchronously go down the gene. And we can measure by labeling the RNA just after we release them, we can measure how far they travel. So this is the data. So this is just to orient you to what we're looking at. So this is an aggregate view of many hundreds of genes that are at least 50 kb long. And this is the Bru-Seq signal we get, just the transcription from this transcription start site and into the gene. Now, if we turn on the red light, the DRB, we effectively block the entry into this gene body. Now, if you wash out DRB and then immediately label with bromuridine, you can see this wave of transcription entering into the body of the gene. And if we allow another 10 minutes to go by, we can see how this wave has moved in to the gene. And by measuring the width of this elongation wave, so this we have three states here. A is upstream of the transcription start site, very few reads. Then you enter this high read region, and at some point you're back to a low level of reads again. So by measuring the width of that, we can determine how far each gene transcribed during the labeling period, which is 10 minutes. So looking genome-wide, or at least on the genes that were 40 kb or longer, we can get elongation rate for each of those genes, and here they're ordered from the fastest gene to the slowest gene. You can see there's a big variation in the rate of elongation across these different genes. We then looked across 5 cell lines to see if cell lines behave differently, and there are small differences, but we did not observe a dramatic difference between the different cell types in terms of elongation rate. So these are the violin plots with the median elongation rate here. You can see it's somewhere about around 1.5, 1.75 kb per minute is the average in our assay. Then looking at genes that are expressed across all 5 cell lines, we ordered them and we're asking if they're fast in this cell line or they're fast in the other. And there is a trend that fast genes in one cell line are fast across the different cell types. And also the slow genes tend to be slow across the different cell types. So we were wondering what makes a gene slow and what makes a gene fast. Is it something with the RNA polymerase that differs, that makes it go fast, go slow? Or is it features of the gene itself that makes it go fast and slow? So we looked at a number of different DNA features, and one feature that came up was an epigenetic mark, H3K79methyl2. So when we ordered the genes in 4 groups, from the fastest 25% to the slowest 25%. And then we asked if we map the different histone marks in these different groups, do they differ? And when we did this for this particular histone mark, they ordered in the order of elongation rate. So the fastest elongating rate genes had the highest level of this methylation mark, and the slowest the least. So there was a correlation between this mark and elongation rate. And this particular mark is put on by a histone methylase called DOT1L that is misregulated in certain type of tumors. So we're very interested in how that plays a role in carcinogenesis and whether elongation rate plays some role in cancer. So we're currently inhibiting DOT1L to see whether the prediction would be then that if we inhibit DOT1L, that we could slow down the elongation rate, if this is correct. So these studies showed us that there was factors that stimulated elongation rate, and this was one of them. And there was some correlation also with 2 other histone marks, the H4K20methyl2 and the H3K4methyl1 had a slight increased level in the faster elongating transcript of genes. You also find that if genes were far away from other genes, they elongated faster. So there's probably some less competition for ribonucleotides, other things that make a gene faster if it's sitting by itself. Also, features of the sequence itself would increase elongation rate. So low complexity was correlated to fast elongation. And finally, if the gene was very long, that tended to make it a little faster. And likewise, we find things that slow down elongation rate. So the density of exons in the transcript slow down. So the more exons, the slower you elongate it. If you had long terminal repeats in the DNA that slow down the RNA polymerase. CpG methylations in the gene bodies was also correlated to slower elongation rate and the density of line elements. And at this point, we don't know exactly why this correlates, but it's been suggested by other groups that exons, when an RNA polymerase approaches an exon, it slows down in order to be able to identify the splice junctions and so forth. And there are probably specific histone marks and potentially nucleosome facing that breaks the speed of the RNA polymerase and it hits the exon. So that was not unexpected. All right, so the final story is with the technique. We haven't published this yet. This is a technique we call BruUV-Seq. And this allows us to find enhanced reads at the beginning of genes, transcription start site, and also we find more eRNA. We enhance for the eRNA reads. At least that's what we think. So I'll be very interested in hearing what you think after seeing what I present. So just to illustrate why we use UV light, and some of you might have seen this animation before, but this is a train map of southern parts of Sweden, where I'm from, and we're going to take a journey from Stockholm to Copenhagen. And if you've been to Sweden, like in Arbor, we have lots of trees. And let's say you picked a day when there was a terrible storm, and the trees start falling across the path that you are going to travel. So if this is a gene, and we shine UV light on the cell, you will get random transcription blockage throughout the gene. So if you were in a train here in Norrköping, you can only travel down to here, and then you have something blocking. But if you're in Stockholm, obviously they don't have any phone or communication, so they just start the 1 o'clock train, the 2 o'clock train, the 3 o'clock, they keep going. If this is a gene, you can just keep initiating the gene, and the expectation would be that you enrich for read in the beginning or near the transcription start site. So is that true? So here again is an aggregate view. So we're looking at genes at least 50 kb long, and they're all aligned at the transcription start site. So this is just the nascent RNA from Bru-Seq. You see reads into the gene bodies. If we now UV radiate right before we label, so we give them 25 joules, which introduce a lot of these breaks, you get a redistribution of the reads. There's less reads in the body of the gene, and that will allow for more reads in the beginning of the gene. And if you go to a higher dose, you see this even more dramatically. This doesn't mean that you have more initiation. It's just that you feed the machine, you give Bob 100 million molecules and he will sequence them. And now the 100 million molecule that you have BRU labeled or mostly comes from here. It's not that they are initiating more. So anyway, this made us think that we can maybe have this as a technique to really point us to where transcription starts. Just first, when you shine something or you give cells something that randomly blocks transcription, the larger the gene is, the more inhibition you will see. So, and this is nothing new, this is totally expected. So the gene size, when you look at the signal you get following UV, it will decrease as a function of UV light. And if you do this experiment, just to look at the technical variability, if you take two samples and grow them in parallel, do everything in parallel, and you sequence them, they become extremely similar. So technically, the labeling, the isolation of the RNA, the library prep, and the sequencing, it's technically very reproducible. So let's look at a particular gene, and we looked at this before, the SMAD4 gene. This is the nascent RNA label we see throughout the gene. If we go to encode the methylation marks, it has one promoter mark. So this should be fairly simple where this gene is transcribing from. And when we do the BruUV-Seq, you see, yes, it's one place that it starts from, and it goes in two directions. This is the divergent upstream transcript. So that was, it didn't help us much. We kind of knew this, but if you have a gene that's a little bit more complicated, like this gene, it has multiple potential promoters. And from the Bru-Seq data, it's a little hard to tell. Obviously, it started here, that's one promoter. But what other promoter was used, it's not very clear. But then when you do the BruUV-Seq, it becomes very clear that it used that promoter, and that promoter. So it helps for genes that have multiple potential promoters to actually find out which one is used. MicroRNA is very interesting. So with the nascent RNA technique, the Bru-Seq technique, we capture the whole primary transcript for microRNA. If you do steady state RNA, all you get is this. This is processed very quickly. But we can map really all the primary transcripts for microRNA. And this is one that is a huge transcript. It will take it like 45 minutes to come up to this 22-mer that's going to be the final product. And just to verify that it starts over there with the BruUV-Seq, we clearly see it has one start site. Now, this was a surprise for us. It was when we start finding peaks outside of genes. So we expected to find enhanced reads at the beginning of genes. But we started to find enhanced reads between genes. And we didn't quite know what to make out of that until we started looking at the encode data, what those sequences may be. So here's a FOS gene. It's highly transcribed. And I'm showing some upstream region here that has a potential enhancer element. We don't see it. So even when we use the nascent RNA, the newly made RNA, we don't see any eRNA. And there's a potential enhancer here that's been identified by other groups. With the BruUV-Seq, you see this enormous enrichment for reads right over this potential enhancer mark. No, these are human fibroblasts. It's a human fibroblast. And in human fibroblasts, the most highly transcribed gene is this thrombospondin gene. It's enormously high. I'm just cutting off this. And upstream of this gene, we see really no signal when we use the nascent RNA Bru-Seq. But when we do the BruUV-Seq, you see this enormous region of what Rick Young would call superenhancer over this large region. It's a 200 kb region. At the time, the superenhancer, when we got this data, superenhancer wasn't a word yet. So we called it enhancer forest. So I don't want to put in a new nomenclature, but this is our term for what we're seeing. So this is under steady state levels, but okay. So here's one more where we look at an aggregate view. So we're taking sequences that encode or calling enhancer regions, and then we line them up from the middle of those regions in either direction. And then we ask, give us all the signal we had for Bru-Seq, for BruUV-Seq with a low dose and with a high dose. And see what do the signal look like. And you can see with just the nascent RNA, you see a little bit of reads over this enhancer. It's not very impressive. But if you shine UV light prior to labeling, you see an enhancement of these reads. And if you go up to higher dose, you see even a higher enhancement. And you can see this divergent character of all of those enhancers that have the middle right there. So it suggests to us that what we're looking at is potentially enhancer elements, and we're capturing this eRNA. We can also look at these enhancers after we do something to the cell. So here we did experiments with normal fibroblasts, just inducing the acute inflammatory response with TNF. And one gene that responds is the NF-kappa B1 gene. So here's control, and then an hour after we added TNF, this gene is very highly transcribed. And if we do the BruUV-Seq, we see upstream of this gene, we see this induction of something we think could be an enhancer element. Again, with the nascent RNA, these sequencing, these transcripts are short, they turn over very rapidly. But we think that with UV light, we can trap the polymerase with the associated eRNA, and that allows us to enhance for these signals. Just a few other examples. So here's a p53 inducible, what Rick Young would call a superenhancer. So this is treatment of fibroblasts with something called nutlin, which stabilizes p53 and turns on a p53 response. And one gene that is affected is this gene PAPA. And you can see this is control levels, and this is 6 hours after adding nutlin. This gene is transcribed very highly. If you go upstream of this gene, there's a large region that also responds to nutlin. So blue here are potential enhancer elements that are giving rise to eRNA, but after nutlin treatment, this is highly enriched, and it corresponds to the gene that is like another 50, 100 kb downstream of this sequence. And finally, I'll show you some experiments we've done with a drug called JQ1. So MYC induces, it's a transcription factor that works closely together with a factor that's called BRD4 that helps MYC turn on both genes and also activate enhancer elements. This JQ1 drug inhibits BRD4 and should inhibit MYC's ability to induce enhancer and induce target genes. So here's one gene that when we looked with after JQ1 treatment was downregulated. And if you look upstream of this gene, there is a region that is highly suppressed by JQ1. And all of those peaks correspond to MYC binding sites. This is from the ENCODE data in these cells we're working on with HeLa. So at all these places where you see a big signal here from our BruUV-Seq, there's a MYC binding site. So these are potentially enhancer elements that are MYC-driven. When we treat with JQ1, we collapse this, and the downstream target gene is also reduced. So we think that with this BruUV-Seq technique, we can genome-wide capture active enhancer elements. And we're very interested to collaborate with people that focus on enhancer to further verify this. So in closing, I've told you about these 4 techniques that we have developed, the Bru-Seq that helps us look at rate of transcription genome-wide, the Bru-Seq where we can follow what happens to RNA over time, so we can look at splicing kinetics of all 200,000 potential introns, and it's a huge signature, and we haven't yet gone through multiple cell lines. We looked at 2 cell lines, and the splicing kinetics varies quite a bit, so that's an interesting signature that is unexplored. And then also we can look at what happens to RNA over time in terms of turnover, and as I showed you, when you look across different cell types, there's a very different setting between rate of transcription and rate of stability to reach homeostasis. So the cells can play with these two variables in different ways. The BruDRB-seq looked at elongation rate of transcription. And finally, the BruUV-Seq, where we can enhance for reads from the beginning of genes and capture eRNA. And just to thank the people in the lab, this is a picture this summer from NCRC. So Michelle has been with me from the start of development of these techniques some 15 years ago. So she's been instrumental in getting these techniques going. The three bioinformatics people that helps me daily is Artur, who just graduated, and Brian, and Karen. So they helped me to analyze all the data. Artur was instrumental in also setting up the analysis pipeline for all the Bru-Seq data. Haley was the undergraduate student this summer that looked at synthesis and transcription, synthesis and stability of RNA across cell lines that I showed you in the beginning. Celine is a graduate student that works, the JQ1 data was from her. Oscar and Masoon was two summer students, and Emily is another technician in the lab. Prasad has worked on the splicing project. And we're open to collaborations. We get calls all the time, and we collaborate with lots of people. I'd like to acknowledge Tom Wilson from pathology, who was very helpful in setting up the pipeline for analysis initially, and still very, very helpful. And these are people we collaborate with currently, and this is growing. It's still not a problem, but you can still add you on there if you want. And with that, I thank you and take any questions. Now we're going to thank Bob too for doing an excellent job with sequence. Yes, Mark.
I'm fascinated by the fact that you show some introns to be retained very long and other introns to spliced out as what we would like. It's when you look at the EST database, at the RNA sequences in the database, there are lots of cases of retained introns in it. I wonder, have you done an analysis of these retained introns in the databases that, at least to my knowledge, were always being credited or misattributed to genomic contamination, whether there's a correlation of these so-called retained, of these things with your retained introns, and so maybe they are not genomic contamination, but just introns that take a long time to splice out.
Yes. We have looked at, so the cell line I looked at was K562 cells, and we've also looked in HeLa cells. And we've looked at other people's published data. I think Tom Gingeris has papers where they fractionated it in different fractions, chromatin, nucleoplasmic, cytoplasmic, poly-A or poly-A plus or poly-A minus, and so forth. And there are evidence, as you said, for introns being retained. Most of that seems to be in the nucleus. There's not very much evidence in the cytoplasm. We haven't yet done a fractionation, but that's on the list, to-do list, to see whether we can, some of the retained one, or they're all in the nucleus and they don't get out. Or, but there's a lot of evidence for retained introns. And interesting with the K562, it's a leukemic cell line. I gave a talk somewhere, and there was a clinician that stood up and said, we do a lot of sample, clinical sample, and every time we do leukemia, we see lots of retention of introns. So they seem to be notorious for being slow splicers or slow turnover of introns, because in the RNA-seq data, there's a lot of intronic signal. Well,
thanks. Do you see any, now you've described with the BruUV-Seq that you're picking up transcription start sites and enhancers. Do you see any classes of RNA signals in there that don't fall in either of those two categories? Do you see anything else?
Well, so we looked at things that are believed to be enhancers, yes, we see enrichment there. Obviously, we see things we don't know what they are. And is that because nobody else has called it an enhancer? Or so yes, it does. You know, there's lots of places, but majority, I think we can of the enhanced are in the beginning of genes and at something that we would suggest would be an enhancer. But there are places where we don't know the answer. Yes.
Okay. Have you compared these enhancer peaks among different cell lines to see how constant were they, or if you have like cell-specific peaks?
Yeah. So, I mean, if you compare cell lines, the genes being on and off are different. So if you look at a particular one that has the corresponding gene is highly expressed, you see a high peak. So if you go to this other cell line where that gene is more or less off, the enhancer is off. So it's a very, wherever we look, the correlation of you know, and we're guessing. We haven't hooked up exactly who talks to whom, but if you look at genes nearby that seems to be influenced by it, it's a very good correlation. If it's high, the enhancer is high. If it's low, the enhancer is low. Yes, please.
Thanks. That was a really nice talk. So I'll just add that there's GeoPet data in K562, so you can make those looping on Paul too. So you can make those looping interactions and figure out who's communicating with who. But I was curious. I don't know if you saw the Phantom 5 paper from Piero Carnici, and they did this cage tag analysis, and they make a big deal that they identify eRNAs too. But to me, when I read that, they looked very different than enhancers that are annotated based on chromatin marks, for example, K27 acetylation. And I thought that your stuff looked much more consistent with K27 acetyl than, for example, the cage tags did. I just wonder if you had any kind of comment or what you think also in DNAs.
Yeah, we didn't know what to make out of the cage. I mean, that came out, we were working on this paper and that came and they very efficiently capture signals from those regions, but they only catch the very beginning of it. They don't know how far it goes. Ours is not perfectly. I mean, we're probably going to inhibit the polymerase from moving, and so we get probably not the whole transcript, but we get a figure that it's at least maybe 2 kb out there. The cage is on steady state RNA too, so you know, and that was puzzling because eRNA turns over so quickly. How do they get such a cage signal? What we're planning to do is to do a Bru cage. So we do the newly made RNA and then do cage on that. And then that should just tell you transcription. Because otherwise, if you do Bru in steady state, it's going to determine, the height of it's going to be determined on the turnover of the RNA, not only on transcription. So, but yeah, the cage is very powerful. It's a very strong piece, but we haven't done the analysis. So you did the analysis and it didn't fit the sort of the enhancer elements from encode code. Okay.
I have a small comment about K562. But K562 has many copies of the chromosome. So how you even think about the spatial aspect? I don't know.
Right. There's a question up there. Yeah. Yeah, it's you. Yeah. Hi. Well, if we do the 30-minute labeling, it's basically gone. I mean, during labeling period, we don't see it. So the trick is to block the polymerase during the labeling, and that sort of builds up the signal that we can detect. So it's so unstable that it may, you know, it's 2 kb long, so it's done in like 30 seconds or a minute, and then half-life might be a minute. So during the labeling, it's only the very last minute or two that's going to go into it that we can capture, and we really don't. We see a little blip, but it's unimpressive.
Okay, thank you so much.
All right, thank you. All right. Oh, okay, that's fine. That's fine. Okay, I should take this off now. So.
Good. Like quite a
Yeah.
useful technique. Convergence of enhancer regulation and transcriptome.
Did you see that paper?
I haven't read it yet.
You know who decided to do this? No? Sister.
Oh, is that right?